University of California, Berkeley, who studies the viruses that infect bees. She's interested in understanding how crop pollination events affect diseases in both managed and non and native bees, and how diseases play a role in insect conservation. Nina is also an artist who uses a range of mediums to illustrate and express her admiration and reverence for the natural world, including pen and ink, watercolor, zines, and digital illustration. To teach the public more about native bees, she has created free California native bee coloring book pages that are available on her website, and we will put that link in the chat. And of course, as always, there will be time at the end to ask all of your questions. So just throw them in the chat as they come up. And then at the end, you can also come off mute and ask. But I would love to say welcome to Dr. Nina Sokolov.
Hi, everyone. Thanks for the lovely introduction. Can you hear me okay? Thumbs up. Yes, I see a thumbs up from Jess. Yeah, it's amazing to be talking to you all today about one of my two of my passions, bees and art. And so I'm going to start today's talk by focusing a little bit about the research that I do in native bees, and my dissertation work, as well as what I'm doing now as a postdoctoral research, and then getting into some of the artsy side of things as well. So as was already described, my name is Nina, and I'm a postdoctoral researcher at UC Berkeley in the Boots Lab, where we study the ecology and evolution of infectious diseases. And I focus on bees as a model system for that. And so we are interested in how pathogenic organisms like viruses are part of ecosystems as well, and how they are impacting ecology. And overall, I want to know how are bees getting one another sick? As my illustration on the bottom shows, one of these ways is that they can get each other sick from shared flowers, as well as so many other roots. And so overall, with my research, the goal is to be able to design management strategies to be able to help bees survive into the future. And by helping pollinators, including managed and wild pollinators, we can help agricultural systems, but also wild areas. And this comes at a time where bees are struggling across the board with honeybees. This past year was the highest recorded losses of commercial honeybees in the US on record, with over 62% of hives lost. This is due in part to viruses, is somewhat of what the early work shows. And so this is very prudent work, especially right now. But it's not just do I not have an aphanus picture in here? Come here. No, back it up. There it is. And so this is also being felt amongst the native wild bees as well. They're also having hard times. Honeybees, even with all this human-mediated management, they're still struggling, let alone wild populations of insects are having a hard time too. So rusty patch bumblebee on the top. This is kind of the poster child of endangered bees. This is the first bee to be listed as federally endangered in the continental US. And I had been presenting in Pennsylvania recently, and so that's why I have the Pennsylvania fact in here randomly. Not found here in California, but is kind of this way towards getting federal protection, and an important insect to be gaining that federal protection under the Endangered Species Act. But here, a more common example that is close to my heart is Bombus occidentalis, what is known as the Western bumblebee. This was, as the name suggests, once common throughout the Western United States, but has declined by 84% of relative abundance since about the 90s. And we think that this is about to be listed as endangered by the state. They used to be very common throughout the state as a whole. And people think that this is in part due to diseases, with this fungal pathogen being a focus of the cause, where we tried to manage bumblebees for indoor tomato pollination. And this was considered a failed management attempt, where after we tried to manage them, they came back from their European jet-setting adventure with some diseases, and since then have been in decline. And so some of my research is in this field now. So I'll talk to you guys a little bit about that. But why are these declines happening? This is for, the common cast of characters that you hear about all the time. It's the climate change, it's the habitat fragmentation, it's the stress, it's the pesticides, it's the poor diet, and the disease, which is what I focus on. And truly, it's all of these different things happening in different ways in different places at different times. But the insect declines that we see is often described as death by a thousand cuts. So it's all of these different components interacting uniquely in different places. And with my research question, I'm primarily interested in what are the dynamics between viruses between managed bees and wild bee species. And so, are any of the declines that we see in managed honeybees and the diseases that we see there, are those having impacts on the native bee populations? And with bee diseases as a whole, just a little biology lesson, there can be transmitted in a bunch of different ways. They can be bee to bee from horizontal to direct transmission. Honeybees, they vomit in each other's mouths to create honey. That'll do it. There's also vertical transmission from mother to offspring. Some of these diseases can happen that way. Environmental, where they forage on flowers that have diseases in them. And there can also be vector mediated, at least in the honeybee, they have this varroa mite that impacts viruses. And although the native bees don't have their own varroa mite, they are impacted by honeybees having varroa mites because it makes the viruses in the honeybees worse and therefore makes potential spillover risks to native bees worse. And how deadly these diseases are, their virulence really depends on a whole bunch of different things. It makes things tricky for biologists like me, where it would be great if things like deformed wing virus, if they had deformed wings, that means that they're sick with a virus. That'd be easy. I could check that off if I notice that symptom. But a lot of these things have sublethal effects that we're not aware of, that we can have a hard time quantifying, and many of them can have asymptomatic infections, and you can't even tell that they're sick unless you do molecular techniques. And so how deadly these diseases are depend on a whole bunch of different things, including the strain of the virus, the dose, how much of the virus that they had, the nutritional status, if they're well fed. And overall, this all combines together to being called condition-dependent virulence, where how deadly the disease is depends on how stressed the host is. And I focus on RNA viruses for a number of different reasons. They can mutate really rapidly. They're an RNA-based life form, and so they don't have these genetic check mechanisms, and they can just like rapidly accumulate mutations. And so when they get into a new host, they can mutate and figure it out. And so a lot of, COVID, for example, is an RNA virus that got into us from some likely rhinophilid bat original host. And so these have some of the highest likelihoods of spillover between species. I'm really interested in these between species dynamics. And in this graph, we can see all these different acronyms of viruses on the x-axis with deformed wing virus, who I mentioned on the most left. Part of the graph where this is an older paper in 2016, so these numbers have changed, but things like deformed wing virus and black queen cell virus have been found, and the green bars are the number of species that they've been found in. And these host ranges can be quite broad. They can be found in ants and cockroaches and other wasps, but we don't really have a sense of what they're doing in these other species or how competent of hosts we're finding them on them and sometimes finding active replication. But we're often finding active replication in creatures that eat honeybees or eat bees. And so maybe they have actively replicating tissue because they ate bees' tissue that had actively replicating viruses. So it's all really I really can't emphasize how early on we are in this field, that we're still finding out a lot of just basic biology of just where are these viruses, who do they belong to, what are they doing? These basic questions we're still trying to figure out. But overall, there's these concern that by allowing managed bees to mix with wild bees, there's a potential for disease emergence via sharing of contaminated flowers. So I talked about a few of these already. Deformed wing virus on the top left, as you can see. And again, these are all called honeybee viruses because they were originally discovered in honeybees. That's where a lot of the money is. But the more that we look into native bees, we are finding these more and more with pretty much having almost no idea what they're doing and what their symptoms can be in the native bees as well. However, with the deformed wing virus, the honeybees seem to be from this Wolfert paper in 2016 show to be honeybee in origin. But there's other viruses like Lake Sinai virus, which is really, really found in honeybees, that we have no idea really what it's even doing in the honeybees, and black queen cell virus, which is also really commonly found in bumblebees, which specifically leads to the death of people that are destined towards being queens. So there's over 31 different RNA viruses and we keep finding more. There's also DNA viruses. There's all kinds of different parasites and pathogens that infect bees. And the more that we look, the more that we're finding. With my research, I have this set in California for a number of different reasons. I'm a postdoc and did my PhD at UC Berkeley. However, when I first got there, I actually didn't even, I had never worked on bees before. I didn't know. I applied with a mosquito project, even, gasp, but totally changed trajectory when I realized, with talking with my PI, Mike, that this is a really wonderful place to be doing bee research for a number of different reasons. And this includes this really high diversity of native bees in the state. We are so lucky to have such a wealth of native bee diversity with over 1,600 species found. And that's because the state encompasses so much of the western side of the continent. And so we have so many ecosystems represented and therefore lots of wonderful bees as well. And in addition to that, there's also this massive agricultural industry that requires this seasonal importation of honeybees for crop pollination. And so one of my major field sites and branches of my research is in the almonds, where over each year in February, over 60% of all honeybees in the US are shipped to California to pollinate most of the world's almond supply. And so what's that doing for disease? We're decidedly, we still are learning a lot about that. But my concern with all of this is, say, honeybees come to the almonds and even though they're blooming in February, so native bees can't really help, even if we were able to restore the ecosystems around the very degraded Central Valley where these are primarily found. Even if we were to restore that, they still wouldn't be able to really be pollinated that much by native bees because they bloom in February. It's super early and it's very bizarre for them to be blooming so soon. And so it's mostly, this honeybee impact that we're worried about. But the honeybees do not exist in isolation. They then go to pollinate almonds. But then afterwards, I wonder if they would pick up diseases there and then would they then bring those diseases to the next sites when the bees are taken from the almond bloom to either another bloom afterwards, or at least around here in the Bay Area and in California in general, a lot of beekeepers can afford to just go to the almonds and then sit their hives somewhere else for the rest of the year and make honey and you do other products. And so there's there's a connection here, right? And so the overall question is, does this mass importation of honeybees into California impact the bees viral community as a whole? So I'm going to spend the science side of this time talking about my field system in the Sierra that I developed with the original assistance of Maureen Page, who's also a wonderful native bee researcher who's now doing a postdoc in Germany. She was interested in the competition side of this story, whereas I was interested in the disease transmission side. And so one of my chapters was focused on bee virus dynamics between managed honeybees and native wild bees in a Sierra wildflower system. And for this, why do I want to work in the Sierra other than it's gorgeous and lovely? Also, it's very scientifically interesting where there's this high bee diversity that's found there. Specifically, there can, be about 20 different species of bumblebees. Bumblebees really like colder alpine environments they do well in. And so there's high bee diversity. There's really lovely high floral diversity. If anybody's spent any time in the Sierra, they would know. And at the same time, there's quite low honeybee abundance from beekeeping not being really that popular, from bears destroying your things, and you put in all this money in investment, and then bears will destroy it. So it's not as popular as areas around where I live right now, and the cold winters kill off a lot of feral hives. So it's a lot lower. There's still honeybees present, but it's a lot lower honeybee pressure than some other areas. It's kind of hard to find control sites without honeybees in the U.S. And this kind of allows for this natural experiment where we can test source sink dynamics or sustained transmission, meaning that with these viruses, we don't know yet if honeybees are the source and they're the main host of these viruses, such that if you were to remove them, the pathogens would die out in the native bees because they're not good hosts. That would be a source sink dynamic where the honeybees the source, the native bees are the sink. And if you can control it in the honeybees, then you can benefit the sink population. However, if instead the native bees are just as competent of hosts, it could instead lead to the sustained transmission where if you bring honeybees in and the native bees are good hosts, even if you were to take the honeybees out, it's kind of too late and they will sustain their own epidemics anyway. So we have no idea which one this is, and I'm sure for different viruses, it'll be different stories. Here's a sweet little bee crawling across a ranger's button. Um, and with a bunch of these are my photographs and my art throughout the presentation. But with this work, I was interested in whether or not viruses are circulating. Pretty much, I worked with beekeepers, the almond pollinators, that then later brought their hives up to these high elevation sites in the Sierra for them. They were doing breeding programs for their bees for this varroa mite resistance and creating wildflower honey at the same time. So they benefited from the low honeybee abundance for that reason. And so I was with this work sampling the native bee community before and after honeybees arrived to these sites. You can place honeybee hives on private land within the national forest, is how this happened. And so I wanted to know, are viruses circulating in native bees before honeybee arrival? Do honeybees bring viruses with them when they migrate? And what are the different dynamics across different viruses? And so with this, and again, I don't really want to spend, I would love to spend a good amount of time talking about the art as well as any questions that you all have. And this is still preliminary, and I'm still working on getting it published. So I'm going to give you guys the cliff notes, little teaser, but stay tuned as more of this work comes out. And I focused on these are the viruses of interest: DWV, chronic bee paralysis virus, and sac brood virus. And these were also my focal bees of interest, where I focused on bumblebees, honeybees, and Osmia mason bees, which are in the family Megachilidae. Um, and that's because they were the most consistent native bee, native solitary bee that was in a different family, because honeybees and bumblebees are both social and they're closely related. And so with some of this disease sharing work, we can see that those that are more related, the hosts that are more related to one another will are more likely to share viruses. But we don't got to get into it too fully, but just the phylogenetic and the relatedness of these species was something that I was curious to see, as well as getting a solitary group in there. So the sampling scheme back in 2021, which is so long ago now, Jesus, this was how I did field work that year, where I sampled in the almonds. I sampled them once when they went into the stopover sites. And then in the Sierra, pretty much everywhere you see a bee was a sampling event. And I sampled the native bee community before honeybees arrived. And then later, once honeybees arrived, I sampled them at 3 time points. Osmia are known for being kind of earlier arrivals, so they weren't there at the end of the season. But I wanted to get these temporal, these viral dynamics through time to be able to get it. And for my focal species, I looked at Vosnesenskii, Vancouverensis nearcticus. Some people call this bifarius. We can agree to disagree. And then Bombus van Dijkii and Bombus flavifrons were my focal species. These were some of the most common ones that were found across my sites. And the osmia, if anybody here has tried to identify osmia bees, you'll know how much of a slog this was. And I totally outsourced this work of trying to identify osmia. They are notorious. And with the help of John Asher, Terry Griswold, and Skylar Burrows, which are all fantastic taxonomists, they were able to identify my collection of osmia, because at each site, at each time point, I took a native bee diversity collection, essentially, to figure out who's here. And we found 31 different species of Osmia mason bees. And so these were in, 20-minute surveys. So clearly there's even more found here. And so this is about a quarter of all North American Osmia diversity almost found here. And this was getting the virus side of these things was totally with the assistance of my wonderful undergrad, Ricky Pankratz, who helped me with processing these buddies. And so my preliminary findings, again, since it's so early on, I want to check some more things before sharing more of this broadly and in recording. So I will just say that viruses were found circulating in the bumblebees as well as the osmia before the honeybees arrived. And this was true across all those viruses that we looked at. And then also honeybees brought viruses with them when they migrated, but it really depended on the virus that we were looking at. I chose three, and each of the three of them had a different story that we were looking at. And overall, I didn't really find any evidence of spillover, which is, yeah, why I'm, making sure I want to be like clear in this story before getting it published, of course. And so, um, overall, the viruses in the native bees that were near honeybee apiaries and near high honeybee abundance were doing essentially the same thing that bees that were not near honeybees or not interacting with honeybees at all. And so everybody just seemed to be kind of doing their own thing, at least for these viruses. Again, different ones will show different stories. And I wonder if this is due to the benefits of a diverse ecosystem. Up there, the density of wildflowers is unmatched. And if they are going to be getting viruses from one another on the flower, then I'm predicting that if there's enough floral resources and they're just not overlapping on the flowers, this can be a way of protecting from spillover, at least between species as much. Because bees don't want to go to a flower that's already been visited. There is all kinds of cues for them to detect whether or not a flower has been visited because pollination's a dangerous job. There's predators, there's birds, there's spiders. So you don't want to be going to a flower that's completely empty. And so they're able to detect if flowers have been visited. There's color changes, there's smell changes that they avoid. And so if there's an abundance of flowers, they can potentially avoid these things, which is a good sign. It's a good these are good results, and I hope that this story continues. And additionally, the honeybees that were brought up here were these Varroa resistance breeders that I was mentioning, and so um, these appear to be, from what I've been finding so far, quite healthy honeybees as far as everything goes, because of this. And so I think it's a nuanced story. But regardless, due to the work that Maureen was finding, where she was finding competition between honeybees and the native bees, as well as me finding the viruses in them, as well as us beginning to find Bombus occidentalis, which you see she's sleeping and not in the little cap when we were doing surveys here. All of this information had the landowners that allowed the honeybees there originally. They said, this seems like a risk. We would rather not. And so the honeybees haven't come back since I think 2022 or 2023, at least with this beekeeping operation. Um, and as we were doing this work, we started to find Bombus occidentalis, which, as I had mentioned earlier, is not doing so great in California. And so it was really special to find them. And we got the necessary CDFW permits to be able to do a project focusing on them. And again, this was spearheaded through a wonderful undergrad at the time, Anna Hatzakis, who is now doing her graduate work at Harvard University. And together we were interested in both the pollen preferences, like what are these creatures eating? And I was interested in knowing what are the viruses of this imperiled bumblebee. And so we found them across 3 different sites, and I got funding to do some more field work this past year. This is a map that shows in purple the historic range of occidentalis versus now you see in black the current predicted range, though I think the actual samples that we're still finding are not much further south than Lake Tahoe, but the projected maps suggest that they can be a bit further. So they still seem to be persisting in the Sierra as well as some of Northern California. But they are okay, and this is just like a little zoomed in picture, but they are doing better in other states. Is the truth of the matter. So in this work with Anna, we figured out a bit of their pollen preferences. So pollenol has very different morphologies that are specific to species. And so if we created this little pollen library, you stain them with this fuchsin jelly, and then create a library of saying, these are the flowers, this is what their pollen looks like. And then you're able to look at the pollen pants of the bees and see what composition of their pollen was composed of which of these different floral groups. And surprisingly, they seem to really particularly enjoy this Parrish's yampa, which is this kind of carrot billiform looking flower that literally I've never seen bumblebees on this thing. It is a bloom that comes out far later in the season. once things are much more dry. And they've got the teeniest little flowers. Bombus occidentalis also have the shortest little cheeks. So I'm wondering if that has something, some connection to it. But they seemed at this time in 2023, they seem to really be enjoying this flower in particular. And again, a flower that I don't really see other bees on. And so this, just my, thoughts, no evidence thus far, but I'm wondering, are they, and thinking about why are they surviving here, and persisting well? Are they able to forage on flowers that other bees are not really foraging on, and therefore are they being protected from some of these disease pressures? It's really hard to prove why things went into decline, and so then proving why something is persisting is a whole nother can of worms. But overall, it was just quite interesting to see this quite different behavior of foraging in comparison to other bumblebees. And so for their viruses, a lot of how much do I want to describe this? I did some meta transcriptomic sequencing across the 4 different sites that I had found Occidentalis at before, to assess their viruses. And everything I have highlighted in blue are all different plant viruses that we were finding. So, whole bunch of plant viruses. One site completely missing any viruses. One on the rightmost one, no viruses at all. The one to the left of that only had one plant virus. But really, the only 2 bee viruses that were found were black queen cell virus and sackbrood virus, in just one of the samples. And so this is quite. And this is the results of that. So only 1 out of 4 sites did any of these bees actually have any bee-associated viruses. And so this is quite interesting. And with this work, this preliminary work, I got the NAPSI grant, which if you all work on imperiled bumblebees, this application for this grant is open and is due in January. And so highly recommend applying for it. This has helped support continued sequencing and another field season to be able to continue diving into this work, because whenever you work on endangered insects or endangered animals as a whole, your sample sizes are really small. And so with disease stuff I'm kind of wondering, are these real zeros? Are they true zeros? Or are they just you sample one? And if it's a low prevalence virus, then you're not going to find them. So doing all kinds of future work to be able to look at the viruses of the other co-occurring bees and use some fancy statistical probability modeling to predict whether or not these are real zeros is the goal. And in my research overall, I really want to kind of highlight this work by Lena Welfert, talking about One Health implications of infectious diseases. And One Health is often talked about in the context of disease spillover from wild animals to humans. So an example being we need to think of human health being connected to animal health, being connected to environmental health. Because things like deforestation lead to more stressed out bat populations. And when they're more stressed out, they shed more viruses and they overlap more with humans, right? And so that is normally the framework that this is thought of. But Lena's group did this wonderful paper about this context for bees as well, and showing how pollinator health is linked to both crop pollination and food production and wildflower pollination, how all of that is connected to environmental health as well as human health through ecosystem services and food security. And so we need to be thinking of this in kind of an all-encompassing way. And yeah, I'll be happy to answer any sort of, research questions along the way, but I kind of wanted to, and feel free to pop them in the chat and we'll be able to talk afterwards. But I want to highlight as well that this term I mentioned and I introduced earlier of condition-dependent virulence makes things like the molecular work for me challenging, but from a management perspective and a land steward perspective, this is actually a good sign. So how deadly these diseases are depend on the condition of the host. So say you have this one instance in which you have honeybees, you have wild bees, and say there are viruses, but there's enough food to go around. Maybe there's an even lower risk that they would interact with diseases at all. But even say if this bumblebee does get sick, if she's not too stressed, if there's not pesticides, she can deal with these diseases. And they have a whole slew of both behavioral and immunological defenses against these things. Um, so there could be the same viral strain, but say in this other instance, whole bunch of honeybees, stressed, pesticides, not enough food to go around. It's the same virus, but it can now lead to mortality in these bumblebees. And so we can help by helping bees be less stressed and giving them the necessary nutrients to be able to mount these immune responses that they have and use their wisdom. They know when they're sick and they can preferentially forage on flowers that have medicine, like they have a lot of wisdom. They know what they need. My soapbox lately is that we don't need to save the bees. We just need to, not, we just need to get out of their way. We just need to let them breathe. We need to let them do what they know what to do. Because a lot of times, and we'll see if people ask me, but people say, oh, should we find vaccines? Should we find these kind of biotech targeted solutions? But I think that the viruses are in the ecosystem. They're not going anywhere. And yeah, with beekeepers, they're already barely making money, And so to ask them, oh, now you have to treat them for every single virus preemptively and they don't have antibodies. And so it's like a continued treatment. It's, these viruses have been here. They're going to keep being here. The reason that they're getting worse is because of the habitat fragmentation, because of the whole ecosystem stress that they're under. And so planting flowers and helping with land stewardship will help. So the latter half of this discussion, well, maybe, I mean, I could take a pause. I feel like I'll maybe take a peek at the chat. But Parrish's Yampa. Yes, that is the flower people were wondering about. So it seems like amongst native bees, maybe only bumblebees are competent hosts, so an overall dilution effect. Bee biodiversity for the win. Um, yeah, with whether or not these bees are competent hosts, so still early on in figuring that out. Um, to me as a disease biologist and thinking about these creatures as hosts, a honeybee's life history is just so strange and so different from other bees. They are alive as a perennial colony all year, 30,000 individuals strong. And so if you're a virus and you're bonking around in that, that's a very different situation than all other bees, in California, that every year everyone dies but the next generation. Two very different life history strategies. And so I'm sure that there's some pathogens that are generalist and can probably infect a whole bunch of different hosts competently. But I think that there must be specialization that exists. And these are just very different life history strategies. So we will see about the competency of the host, but you also brought up dilution effect, which is this idea of with increased diversity, there can be decreased disease. And so that seems to be happening sometimes, other times not as much. Whether or not interaction between diversity and disease is positive, it depends on how general the viruses or pathogens are. If they're super generalist and they can infect a bunch of different things, then it doesn't matter to them whether or not it's this species or this species. It's just more susceptible hosts. So it can go the other way and be called an amplification effect, where with increased diversity there can be increased disease. So it's all over the place, complex. I like dilution effect. It feels right to me, where I think that the benefit is that with it, I think diversity is the answer, is one way or another. And I also see Jess mentioning that the California State Beekeepers Association published a white paper to aid in lobbying efforts to push allowing commercial honeybees onto public lands. Yes, I saw that as well. And I think that there are needs amongst honeybees to improve their nutrition, to improve their health. But I think that state lands and federally protected lands are there to protect native biodiversity. To me, that's what I understand the point of them to be. And I think instead they should put efforts towards, we have so much land dedicated to agriculture. Can we not just, focus our energy towards, as this honeybees are an agricultural animal, to, improve agricultural habitat for bees. So it's not just one bloom and then there's nothing, but to have more polycultures, to have more other blooms at the same time, or sequentially at least, so not just have one bloom and then nothing. And then I see one more question from Katrina. Do pollen or nectar from our native plants contain any antiviral compounds that may help native bees fight infections? Yeah, that's a whole tangent that I've been finding quite interesting. And if you like those kinds of stories, right, Doctors by Nature is a book that just came out from a disease ecologist named Jaap de Rood. And he is a whole book about these kinds of stories of animals self-medicating themselves. And the more that we're looking, the more that we're finding it. Thus far, it seems like a lot of the evidence for the medicinal behavior has been for parasites more than viruses. And so sunflower pollen has been shown to be medicinal for bumblebees against this gut parasite called Crithidia. And that was work done by a person with a biochemistry background. And so she was really being like, what plant secondary metabolite is this? But they found in the end that it was just the spiky shell of the pollen. Aster pollen is like very, very spiky, spiky, spiky little ball. And it seems like that just the physical structure of the pollen seems to induce an immune response and help them clear parasites, both in the lab. And then when they looked in the field, they found lower parasites in bumblebees at sites that had higher sunflower plantings. So both in the field and the lab. And so aster pollen seems to be, as a whole, not just sunflowers, seems to be beneficial. And then what was it? There was another flower. Gordon Fitch up at York University is working on. Ooh, it's another garden flower. Okay, after we're done and we go into a little bit more chats, I'm going to look into that because the name eludes me. But yeah, I think that the more that we look, the more we're going to find. And so when you're planting things in your garden, think about you're planting nectar flowers, you're planting pollen, protein rich flowers, but also think about the medicine side of things. So my pet theory that I don't think has any evidence is yarrow as well, because I see bees like sometimes on there, but most of the time not. And yarrow is also really medicinal for humans. So yeah, super interesting topic. Also shows the benefits of diversity. So that's a really great question. I'm going to continue on with the art side of things, but please feel free to keep asking questions in the chat as we go, but we'll also talk at the end. As in the introduction was noted, I'm also an artist. No formal training at all, truly just hanging out with weird goth art kids when I was growing up, and that's how we would hang out and spend time together. That was my training, as well as YouTube and just practicing. But I find it a really, really wonderful component of my life that lets me get to know these creatures more deeply. And art helps me with my science in a number of different ways where it improves my taxonomy skills. I always say that it gives me the eyes to see, because if you're staring at some of these examples, I was staring under a microscope at this thing for hours, and so the more that you're looking, the more that emerges for you. And so it's these observational skills and trying to communicate what you're seeing onto a piece of paper allows you to see more. It also helps me communicate complex ideas. In papers, I'll show you some of my art that I've done in publications, but also in presentations such as this. And my posters, I always liked them being aesthetic and beautiful. As I was saying, that enhances my observational skills, which can inform my hypotheses. And overall, it just helps me get to know these creatures more intimately and helps me fall in love. Insects are my muse. Here on the right is an illustration of an osmia bee getting infected from the flowers that she forages on and giving it to her offspring. So I did get a little I said I wasn't formally trained, but that was, a little bit untrue. I did get training at this wacky building called the Royal Ontario Museum, where I did my undergrad at the University of Toronto. This was our natural history museum there. And when I was working there, there was camera lucida, if anyone's ever heard of that. It's a microscope attachment that essentially acts like an arm off of the microscope with a series of mirrors, such that when you look into the microscope, you there's a series of mirrors that you can see both your specimen and the piece of paper that's next to you at the same time. And that allows you to get, it's not a very great, resolution image, but it gets you perfect proportions. And so there was a PhD candidate at the time named Julio Rivera who was redoing the whole praying mantid phylogeny at the time, which apparently was a mess. And he was illustrating and describing new genera and things like that. And so he was my teacher in the scientific illustration world, which I had been, as I said, just no formal training, do whatever I want kind of art style. And then learning biological illustration, it is, that stuff's hard because it has to be perfect. And so he would call me out. He would say, "Is this patch of hairs here actually?" And I'll be like, "No." And be like, "Okay, well then it shouldn't be in there." So it was a big skill improvement to be able to go to this museum and across month periods be building these illustrations old school style, like in layers, in ways that, yeah, were challenging. Here is Chrysatoxum flavifrons, which is a wasp mimicking hoverfly. As you can see, all the little stippling, all the little dot details, that took forever. This is all pen and ink illustrations. This is not the specimen that I was using originally, but as you can see with museum specimens, all their little legs are like different angles. And so with this, you would get them in the perfect position for one leg, and then you have one sheet. Then you get them in a perfect position for another leg, and then you'd have that on another sheet. And in the end, you're layering all of these different sheets over each other to get you the perfect specimen. So it was a lot of work. And here, this wacky creature is an ant mimicking tree hopper with this crazy projection off of their pronotum to look like an ant. Again, these are all the stippling based illustrations that really honed my skills. This is Bombus affinis, who I had a friend commission me for this before I started my PhD, before I even knew that I wanted to work on bees, very fortuitously. And so, as I mentioned before, here's the poster child of endangered bees with the little middle, not in color, but the middle dark bit on their abdomen is a rusty color, as the name suggests in real life. This is what they look like. So pen and ink is kind of one of my favorites styles. I like the control of it. Watercolor as a whole um, is a practice in letting go of control. And I also really like digital. I did this illustration from under the scope during the bee course, if anyone's heard of that. 2 weeks in the Arizona desert with a whole bunch of beaks. That's where I originally met Jess. And had a wonderful time. So this is a squash bee, and for those that don't know, the crazy projection of the mouth, that's what their tongues look like, which is wild. And squash bees are specialists on squash, as the name suggests. Yeah, here's an image of the crazy tongue sticking out. These were some markers. Yeah, I was using alcohol-based markers for a mason bee, a sweat bee, agapostemon, and a megachile male bee as well. So these are all just examples, as I was saying. But with bees, there's also the flowers, and so having botanical illustrations is a fun addition to my observations and everything too. And if anybody here has ever done nature journaling at all, that's a really wonderful practice to um, to slow down and to observe and to be in a place and to remember moments, from these illustrations, like those memories are locked in my brain in the long term. And this is more of an act of trying to let go and not be perfect, because these things you're sitting in the dirt and you're uncomfortable, and you can't be, comfortable at your desk with working for hours, the sun's beating down or the bee's already flown away. And so this is a very different practice that, if people are curious about, would love getting into. But nature journaling is a wonderful movement. The people who do it are great. They're a bunch of nature-loving art dorks, such as myself, and the communities that are formed around this are really wonderful. This is very unreadable, but that's just digital illustrations. And I mentioned that I do illustrations for my papers. So here's a, kind of more graphical abstract of looking at the impacts of varroa. Like I said, if honeybees are covered in varroa, and even though that the native bees don't have their own varroa mites, they can be impacted by having worse spillover risks than with honeybees that have low varroa. So always trying to emphasize to honeybee keepers to control their varroa mites due to this shared disease pressure. And so this was one of my figures in this Proceedings B paper that came out for my dissertation. Math has always been scary to me personally, and so which is hilarious because I'm in a mathematical modeling lab, but I've gotten less scared through time by realizing that a lot of these equations for mathematical modeling are just stories and are just sentences. And so I used that structure to kind of and illustrations to break down these are what all these different scary looking math parts mean and alongside what the biology is looking like at the same time. So again, trying to emphasize translating complex ideas into simple ones. And here for the native bee people, this was a wonderful collaboration with a at the time graduate student named Stefania. This is her paper in the top right where she was quantifying the behaviors of a kleptoparasitic bee, so bees that lay their eggs in the nests of other bees. So this is a Tripiolus with a xenoglossa as the host, and she characterized this kind of behavior that they do. And she gave me little video clips to be able to illustrate this sequence of behaviors of they do a little flyby that can either lead to that they leave or they do a little hover to check it out. They land, they got to inspect it. So they do a little antonation and then some amount of time they depart and they say, nah, I'm out of here. If somebody's home, or in some amount of time they enter. And then when they enter, they then exit, and they can either depart immediately or they do this kind of pre-departure groom, where once they're out of there, they want to clean themselves up. They do a little soil shuffling. So as I'm watching these like little very small videos, I'm trying to get to illustrate these behaviors. So that's, the science side of things, but I also really like this for outreach. I've worked in Bay Area scientists in schools doing illustration workshops, and over the pandemic, and I highly recommend this for you all too, if you're ever teaching about native bees, and I was teaching kids about, how you can steward land, and wanting to teach them art skills too. So this was all over the teaching kids over Zoom during the pandemic. I really empathize with teachers. I did that for just a short period of time, and I'm like, man, that was hard. But this is a little project that we worked on where I was suggesting it to be an illustration, but in the end, some kids did dioramas. I forgot dioramas existed, but it was so cute. But I suggested having picking a favorite flower. So you want to have different things that bloom at different times in the year. So picking a spring, a summer, and a fall flower, as well as picking nesting materials to illustrate, and then picking a favorite bee. And so doing this like design your own little native bee haven, where you draw your favorite bee, the flowers that they feed on through the year, and their nests, and kind of have little reflections on it. So that was a cute activity, but again, dioramas emerged from that, which was amazing. I then developed this native bee coloring book activity in collaboration with CLEAR, which is Communication, Literacy, and Education in Agricultural Research. And this, again, was a pandemic project of trying to do an event for the Bay Area Science Festival. In which I wanted to have kids either be able to draw along with me, or if parents had printers and could print these things out at home to learn about native bees. And so I did an event where I did this drawing of a Melisodes, as you see on the left, in 2020. And then there's like a little cute little contest where they submitted their art on the right. And this has really evolved as an activity overall, where I'm using the art skills to be able to illustrate these coloring book pages, which for me helps me remember what these creatures look like, but then also creates this activity that I can hand out essentially as free as it can be. Like I have access to a printer in comparison to stickers or t-shirts or other things that you hand out. I think it's a really fun event. So here's me and my lab mate Sarah at one helping with the Lawrence Hall, who've been really instrumental collaborators. And now that has emerged into this wonderful collaboration with Vincent Medina and Louis Trevino at the Atoya Initiative. They are both Ohlone leaders who are the native people of the Bay Area. and have been doing a lot of work of bringing in Indigenous science into the Lawrence Hall of Science as well as at UC Berkeley. And Atoy is the Chochenyo word for describing a philosophy of intention of repair and mending, which is really critical to think about when in collaboration with Indigenous communities as a Western scientist, and specifically one at UC Berkeley, because UC Berkeley um, has a storied history with this group of people, including um, saying that the Ohlone people were considered culturally extinct, and so Ohlone isn't the Ohlone community isn't a federally listed tribe, in part due to the declaration that was made at UC Berkeley. So the fact that they want to collaborate at all is a big show of trust, and I hold that very tenderly. And so together we've been working on doing plant pollinator surveys to figure out what native bees and lepidopterans are visiting their gardens that they're stewarding. I created this little logo that you see in the middle. And on the bottom right, you see me and my lab mate, Signe White, who is a moth expert. And together we've been doing native pollinator surveys at their gardens as well as other native gardens. That I won't get into, but we were doing non-lethal sampling and doing photograph-based surveys to pretty much be like, who's here, let's get to know the creatures that are visiting these flowers. And together we've made some native bee coloring book pages that include both the native bee wisdom that I can help provide, as well as the Chochenyo name for some of these plants, the hinai, for the Yerba Santa specifically, and the ways in which these plants are culturally important for the Ohlone people. So trying to blend and share knowledge between each other and share what creatures are visiting their gardens. So I think that the hosts will share a link with this, but there's a little QR code here, and also I'll have it at the end to my website where I post these. Please share them widely. I feel I like spread them. If you color them in, I love seeing emails by them. They really make my day when people do them. And as I create more, I'll keep posting them on my website. And so think that that's plenty. I'm going to thank the Boots Lab members that have helped me along the way and the funding resources, folks at the California Department of Fish and Wildlife, Hillary, who I've seen on this call before, for all the help and along the way, as well as with the field research, the UC Natural Reserve System. Thank you to the landowners and the beekeepers, and of course, thank you to the bees that had to be sacrificed for some of this research. I hold that seriously, and so I want to make sure to tell their stories and to advocate for them. So thank you all for your attention. And like I said, here's the QR code for any for the link to my art website. And yeah, with that, I'll take any questions.
Thank you so much. So